So what's the polarity of this nucleotide sequence? Starts on the left side with 5 prime. And then 3 primes on the other end. So for PCR to work, or for DNA replication, because it's the same thing, PCR, or the actual process of replication that happens in our cells, it's the same thing. The DNA polymerase enzyme is going to add nucleotides to which end, for example, if this was being replicated right now. You'd add another nucleotide on the 3' end. So what we need to have DNA synthesis is you have to have a free 3' end. So for example, now this is not the answer, but that could be a primer sequence that would allow replication of the opposite DNA strand. Its 5' end would be there, because DNA is anti-parallel, so you always have a 5' end next to a 3' end. And that means this is a primer that could be used, and DNA polymerase, the enzyme, could come recognize that 3' end, and it could move that direction and add in all of the complementary nucleotides, a G, and then an A, and then a T, and then a C, and a G, and so forth, and keep moving and replicate the other strand. That's the point. DNA replication, PCR, same process. So with the answer that you gave, the primers that you chose, you said one of them was GATCG?
I just copied
No, go ahead.
exactly what that thing was.
Yeah, so let's see.
I said CGATC.
We're trying to amplify the central 10 nucleotides. So the question is, as you already know, what's the primer pair that you need to copy that part of this DNA sequence? So where would you put the 3' end, just like we have here? We've got a 3' end that's going to start, it's going to define one edge of the new section of DNA that's being replicated by our DNA polymerase, RNA polymerase. It's DNA polymerase. So where would you put a 3' end that would copy across that 10 nucleotides?
So would you start before the 10? So like G, A, or well, yeah. G, C, like right below. Would you start here? Those?
Right. So if you had a primer sequence here, what would it be? What would the nucleotides be?
Starting at the C.
Yeah, let's start there. So
G,
there'd be a
C,
G, C, T, A. And if we were going to make it 5 nucleotides long, we'd add a G there. So you've got the complementary sequence to the strand, which means that you've got, it's anti-parallel, so the 5' ends on the right side, which means, again, you're absolutely right, you've got that free 3' end there. So polymerase, DNA polymerase, is going to bind there, move that direction, and fill in those 10 nucleotides, plus it's going to keep going down the chromosome. But the important part is that you've just synthesized that 10 nucleotide stretch that we're trying to synthesize.
So I could have chosen this side too.
Well, you have to. For PCR, you have to have a primer pair. Because, so here's the, and we're going to talk about this in class, but I'm happy to talk about it here. That original double-stranded DNA molecule that we're trying to copy has 2 strands. I've given you the sequence of 1 strand in black. But what you have to realize is it's double-stranded DNA. So there's another strand down here that's the complement. TCAG, TCG, ATA, and then there's whatever's in that black box, and then the rest of these complementary nucleotides. So right now, from the top strand, you've made this primer, and it's going to synthesize a bottom strand. That's how so now you have a double helix here, but you've got this other template strand that you need to make its complementary strand for, to go from 1 double helix to 2 double helices. So for the strand that I gave you initially, this strand that starts the 5' on the left and goes to 3' on the right, we've got the 5' on the right to the 3' on the left strand that we also have to make a copy of. So where would you put its primer that would synthesize across that?
So the primer of that is really just that, right?
Exactly. So there's the key realization, is that that 5 nucleotide primer you're going to pick is there, and the complement of CGATA is GCTAT, which is exactly the sequence that's on the top strand.
So one of them is going to be like already there, basically given to you.
They're both given to you. Yeah. So I'll give you one strand. This next step you should always do for these is to write out the sequence of the other strand. Those are your two template strands. So I'll give you the top strand. You write out, this is the other strand that's complementary. And then I design the primers to be complementary to those sequences. And as you just pointed out, the primer sequence is going to be identical to the sequence of the strand that you're not using as a template strand. So over here on the right side, if we wrote out that DNA sequence that's complementary to the top strand, it would be GCTAG, which is exactly the same as the primer pair we designed for starting the copying of the top template strand. So, to summarize, maybe this is a summary, the primer sequence is always going to be present in the sequence of the strand that's the one that you're not copying. Maybe that's not a good summary.
Anti-parallel strand.
Basically. So if you're trying to copy the top strand, the sequence of the primer is going to be in the bottom strand. And when it's the bottom strand that you're trying to replicate, the primer sequence that you're going to use to replicate it is part of this sequence in the top strand. And that's all because both have to base pair. So if we're talking about copying the bottom strand, we know that the top strand has a complementary sequence, and it's anti-parallel. That's exactly what we're looking for when we design a primer. We want it to be the complementary sequence, and it needs to be anti-parallel.
So the two primers for this sequence are there.
Yep, GCTAT from the 5', so you make sure you write out the orientation. That's 5' and that's 3'.
And then up there.
Right, so we've got G, so reading it in proper orientation, GATCG.
But you could write it like that, 3 to 5, like on the example.
Yeah, if you drew it out, as long as you've labeled the orientation, so I know that you know that that's the orientation, that's fine.
Okay.
Can you circle those two answers in like another color?
Sure.
So if I go back to it,
Yep.
like know what
Let's see,
the
blue.
answer was.
Yeah. And I'm recording this, so I'll post this on YouTube too, in
Okay.
case you want to go back and listen to our conversation or see what we've drawn.
Okay. Thank you, that explained.
So the other tip I have, again, to reiterate, if you get one strand of DNA sequence and it has anything to do with designing primers, I would write out the second strand, a complementary strand first, so you have both of the template strands like I did here in black. And then you have to remember that you need two primers. One is going to synthesize from left to right, like the bottom strand here. DNA polymerase is going to bind there and synthesize the second strand of the bottom template, and you're going to have the primer that is pointing, 3' end is pointing to the left to synthesize the second strand for the top black or template strand. So that's why we refer to primers as they always come in pairs. You design a primer pair because you have to have two different sequences. Those two primers have different sequences. You need them both to make copies to generate two double helices from the one that you started with. you started with a template molecule that was 2 black template strands. I only gave you the sequence of the top strand. So, as we learned with the Messes and Stahl experiment, when you separate those two, then you get primers annealing. In the case of DNA replication, that's primase that makes RNA primers. But in the case of PCR, those RNA primers then are used to synthesize, let's say, that direction, and that's how you get 2 double helices, 1, 2, when you started with 1. So the only way to complete DNA replication is to have 1 primer there and 1 primer there.
It's like anti-parallel.
So it's still anti-parallel. Yeah, you generate 2 anti-parallel DNA double helices from the one you started with. Okay, yeah, so this was a microlecture video question from, I think, yeah, the Agarose gel electrophoresis. Maybe it was the PCR genotyping video. Okay, so the first question was, where is their heterozygosity?
And the C and G.
So we've got here. So this is the DNA, this is the 2 chromosomes in one individual, right? The chromosome 1 they got from their dad, the chromosome 1 they got from their mom. There's heterozygosity, which means they have different nucleotides at the same position on the chromosome. Dad's a mutant or mom's a mutant, well, who knows? But they've got a different DNA sequence. So if you want to amplify this DNA fragment from both copies of this chromosome, what sequences would you use? In this case, the answer is a bit odd, but we approach it exactly the same way we just did in the previous example.
So just look to the right of the...
So each of these is going to be 5' to 3' orientation. And we could write out, just for one of these, I'll write out the other strand. So that's the other strand. So if you want to amplify this entire sequence of DNA, the whole thing, which is probably 20 nucleotides if I counted correctly, yeah, it's about 20 nucleotides. So if you want to amplify the whole thing, you have to have primers that go all the way out to the very end of the sequence. And you want a primer that has a 5' end there and a 3' end that will let you sequence or replicate DNA into that missing part. So what would the sequence of that primer be? Right. Except, we want this to be complementary, so this is the 3' end. So we need a primer that's going to be complementary to it, so it will base pair with that bottom template. Yeah, so we need 3 A's, maybe, and 3 T's. If you were going to make a 6 nucleotide long primer. So that would let polymerase come in and add in what's complementary to that bottom strand sequence. So then it would synthesize a C, it would connect it to another C, and then some G's, and some A's, and some T's, and then polymerase would have made the second strand from that bottom template strand.
And that new primer is starting at a 3' end, right?
So we've got one primer now that what colloquially geneticists refer to as a forward primer, because polymerase will copy DNA in the forward left to right direction. So that'd be a forward primer. It synthesizes from one template strand. So now we need a pair of primers. We need the second one, which is not going to use the bottom template strand, it's going to use the top template strand. So we need something that goes (5' to 3'). We need a primer that will stick onto the top template strand by base pairing. So just like the previous example, we have two template strands here. We have a template strand there that goes 5' to 3', and we have the one on the bottom.
So this is incorrect. I'm writing 3' here.
That's
This is my primer going towards 5'. That's wrong. This
That's
should be 5.
well. So, two issues. One is that so you have the polarity correct. If that thing you circled in blue, those 6 nucleotides, if that's a
the
primer,
Yeah.
which direction is that going to synthesize DNA?
I thought it was going towards this way.
So where on a primer does RNA or DNA polymerase add the next nucleotide? The 5' end or the 3' end?
So it is wrong.
Right.
Okay.
That's incorrect. And the reason why is because that primer. So what you didn't do was you didn't label the polarity of the bottom strand. So label the polarity of the bottom strand.
So this one is 3'.
Correct. Exactly
So
right.
if I would have done that, I would have known it would have kept going this way.
So the primer you have written here, AAATTT, doesn't do anything with the top strand. It's going to base pair with. You've written the complementary sequence to the bottom strand. A pairs with T, A pairs with T, T pairs with A. So all you have to do to make that primer work is now you have the primer orientation polarity labeled wrong. Because you never have a 3' end base paired with a 3' end. So all you have to do to fix the problem is
Make the arrow go this way, right?
switch the polarity of the primer. So that's its 5' end. That's its 3' end.
Oh, so it still is going this way.
So yes, when you get the polarity right,
Yeah.
yes. When the 3' end's on the right side, then it will be a primer that will synthesize to the right.
So this entire thing is the primer to the first, to the DNA sequence, this whole thing.
So you would start with that.
The 6 nucleotide long primer. But you said that you wanted the whole thing.
So that's what DNA polymerase, what's circled here in, what is that, yellow or green? Or yellow-green, whatever. Colorblind, help me out.
Are you really?
Yeah. Not that badly, but if it's got a green component, I wouldn't be able to tell you. Yellow, green, and yellow both look yellow to me. So what's circled here is the initial primer. So that's the piece of DNA. It's just a single-stranded piece of DNA that we purchase from a company. We say, send us that 6 nucleotide long piece of DNA. And then what's written in yellow is what DNA polymerase has added. That's newly synthesized DNA. Where it grabbed onto the 3' end of the primer, and then it used that 3' hydroxyl group to add the next DNA nucleotide onto. That's all the primers are. They function, which may or may not help. I apologize if it doesn't help, but they function exactly the same way as primers do during DNA replication in our cells. In our cells, that protein primase builds a primer so that DNA polymerase has a 3' end to start replicating our DNA. In PCR, which is something we do in a test tube, really, not in our cells, it's the exact same process, except we don't use the enzyme primase to create primers. We want control over what the primer sequences are. So we synthesize, company, in Oklahoma in this case, synthesizes those short pieces of DNA. We put them in the tube with some DNA polymerase and the template molecule. And so the primer base pairs with the template strand. Polymerase recognizes that 3' end, and then it starts adding those new nucleotides.
Thank you, that helped. It's a good, model.
The overview, yeah.
Wait, so then what's our second primer?
So we haven't done the second primer yet. Exactly. That's going to go up here.
So that one would just be...
So we need a primer that's complementary to the top strand.
And that one would be AAATTT.
Right. And in this case, I really want to... yes. And that's something I inadvertently did, which was not a good idea. Let me do this in black, too. But the complement to TTTAAA reading here from right to left, the sequence of that primer is going to be AAATTT. It just so happens, in this case, that those two primer sequences we just created are identical. Right? This primer was AAATTT, and that primer is AAATTT. That's totally by chance. That will not always happen. Normally the two primers will have totally different sequences. Of course, the reason why is because I don't know if I hope I didn't do this on purpose, but this DNA sequence is a palindrome. So you notice the top strand is AAATTTCCG, the bottom strand is AAATTTCCC in this case, because that's where that mutation was. But I shouldn't have written a palindromic sequence to amplify with PCR.
So the two primers here are there, 5 to 3, and then 5 to 3.
Right. So the final primer sequence is all circled in blue. And they're both, starting at the 5' end, they're both AAATTT. They don't have to be 6 nucleotides long, by the way. I will usually specify how long to make them to make grading easier on me. But the point is that I shouldn't care about how easy the grading is for me, because, again, depending on how I write the question, there might not be one right answer. So if I said, for example, for this bottom strand, I could say create a primer that you could use to replicate that bottom strand. Here. You could have picked a 6 nucleotide long primer and it would have been AAATTT. It doesn't have to be 6 nucleotides. You could have picked the first 4 nucleotides. So your answer could be AAAT, which would be just as useful because there'd still be a 3' end there on that 4th T that DNA polymerase would grab onto, and it would synthesize T, T, C, C, G, 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 and so forth. So there is a correct way of going about designing primers, but in this case, there isn't necessarily a specific nucleotide sequence that will only be the only one that works, for example.
On the exam,
Yeah.
all of the case studies, will there be questions about them?
Not necessarily. I'll probably avoid some of the case studies because we didn't talk about them in class. So I feel very strongly, although I might make a mistake and accidentally put an exam question in that deals with something that I thought we talked about in class, I always do my best to try to avoid doing that. But my goal is only to use things in the exam that we've actually spent time talking about in class. So some of the case studies we didn't talk about in class at all, I'm going to try my best to avoid asking any questions about those.
Because we only really talked about 2 or 3.
I think it was 2, maybe 3. There was the one, I was trying to tally that last night, just did from memory. There was one about genotyping oocytes by extracting their polar bodies and determining what chromosomes were in the polar bodies to figure out if an oocyte, the primary oocyte, the egg, would be missing a chromosome or have an extra chromosome. That was one we went through a bit in class.
That was like the 1 + 2 + 1 = 4.
Right. Yep. And last class we talked about the allele frequency tables and calculating the probabilities of people having different combinations of different alleles. So from memory, those are the two that would be most likely to show up on an exam.
I know that in that case study we were like looking for aneuploidy. But and you kept doing it over and over. I just didn't understand like how you're adding up the numbers and where you're getting them from.
So we could just look at a simple case. So this is based on, I think the first author's last name was, oh, it wasn't, oops, how it all. So the x-axis, they were looking at number of chromosomes. And let's just pick one, the y-axis was number of chromosomes, the x-axis was which chromosome they were looking at. So let's just pick one chromosome for now to talk about. And the three different graphs were something about looking at the first polar body, the second polar body, and then the oocyte itself. We're doing time. We've got to leave in a couple minutes. Okay. Based on how meiosis works, you get a division, the first division generates the first polar body. So that's polar body 1. And after the second division, you generate the primary oocyte, which is that big cell, and the second polar body 2. That's the origin of all those cells. You divide once, that's the first polar body and what will eventually become the oocyte. The second division, you get the second polar body, and then the oocyte itself. And here's where the addition comes in. Meiosis always starts with a cell that has 4 double helices. So you divide those in half at the first division. So the first polar body should get 2 copies of every chromosome, double helix, let's say, and 2 go there. And then in the second division, this cell divides its 2 copies of that double helix into the 2 daughter cells as well. So you wind up getting 1 copy of the chromosome in the second polar body 2 gets 1. The first polar body still has the 2 it got in the first division, so nothing's changed there. Polar body 1 stays the same, and then the oocyte has 1 copy. So at every step, we've got 4 double helices, that's 4. We've got 4 double helices, which is 2 + 2, and we've got 4 double helices, which is 1 + 1 + 2. No new DNA is synthesized, no DNA is lost. It's just being divided up among 3 different cells. You have 4 double helices, 3 cells. You can only divide it in a handful of different types of ways.
Thank you.
Yeah. So, as an exercise then, if this is chromosome 1 we're talking about, where we start with 4 copies, it divides into 2 and 2, and then 1 and 1 and 2, you expect to see, let's see, first polar body we said has 2 copies, so we expect in blue that all of chromosome 1 there should be 2 copies of every base pair from left to right. There should be 2 copies at every base pair of chromosome 1 in the first polar body. That's what we expect, just based on how we know cell division and chromosome segregation occurs. And in the second polar body, there should be 1 copy of chromosome 1 from its left end to its right end, and there should be 1 copy of chromosome 1 in the oocyte, from the left side of the chromosome to the right side of the chromosome.
At the top, it's at 2, not 1.
That's, yeah, so that's 2, 3, 4. Yeah. So what that manuscript showed was that if you actually took a real first polar body, second polar body oocyte from a female human, then you might see something like that. So this part of chromosome 1, there are the expected numbers of copies of that part of the chromosome, and the same is true over here. But in the middle of chromosome 1, for whatever reason, the first polar body has 3 copies of chromosome 1. And there are 4 total copies of that, of every part of chromosome 1. So if the second polar body just has 1 complete copy of chromosome 1, no extra, no missing, you should be able to predict what are we going to find when we look at the DNA content of the oocyte.
Should there be
Missing.
So where is there none? So there are parts of this, so every part of this, when we look in a column vertically here, when we add up the total number of chromosomes, it has to total 4. So in this section on the left side of the chromosome, there's 2 copies there, there's 1 copy there, which means there has to be 1 in the oocyte.
Wait, why'd you say there's 2 there and 1 there?
So the first polar body has 2 copies of the chromosome, the second polar body has 1, so that means there's only 1 left, and it has to be in the 3rd cell. And the same is true on the other side, where the pattern's exactly the same. So the question is, what's happened in the middle of the chromosome? We've got 3 copies we've discovered experimentally just by looking at the copy number of the chromosomes in these cells. Instead of 2, the first polar body has 3. Here, we expected the second polar body to have 1 copy. It does have 1 copy. So, right, there have to be 4 copies of this chromosome, so that already totals 4, which means there is none left for the oocyte. So in this egg, from this female human, there was part of her, in the egg that's going to be fertilized and make a human, it's missing part of chromosome 1.
And that proves aneuploidy.
So that would produce an aneuploid offspring for that part of that chromosome, which is probably something that would lead to some sort of death or mutation, or most likely something like